Everyone, good morning or good afternoon. You might have seen there that we will be recording this meeting. So if you're all right with that, thank you. And if not, please continue to keep your cameras turned off. So for today, we have two lovely trainee speakers that I'll be introducing in a moment. But first, just a couple of reminders on behalf of the CSMB. So our two trainee talks will be 15 minutes each, followed by a 5-minute question period. And with that, we would like to ask that if you have a question, use the raise hand function, at which time myself or Matt will unmute you, or you can place your questions in the chat. The seminar series is brought to you by the CSMB. And if you're interested in becoming a member or learning more about what we do, feel free to visit the website here. And without further ado, I'd just like to welcome our first speaker today, Dylan Farnsworth, who's a PhD candidate in William Lockwood's lab at the University of British Columbia. And today he'll be presenting his work entitled MEK Inhibitor Resistance in Lung Adenocarcinoma is Associated with Addiction to Sustained ERK Suppression. And with that, I'm just going to let Dylan take it away.
Thanks for the intro, Kathy. Yeah. Can everybody see the screen and hear me all right?
Yep.
Okay, thanks. Okay, good morning, everybody. Yeah, like I said, my name is Dylan, and today I'm going to be presenting on my work entitled MEK Inhibitor Resistance in Lung Adenocarcinoma is Associated with Addiction to ERK Suppression. So I'm from the Lockwood lab here at UBC, and we study the main area of our study is lung adenocarcinoma, which is the most common subtype of non-small cell lung cancer. And one of the main characteristics of lung adenocarcinoma is the detection of frequently mutated oncogenes. So on the right here, I'm showing a pie chart of some of the most commonly detected ones in patients. And I'd like to, our area of study focuses on mutant EGFR and mutant KRAS, which are the two most commonly detected driver mutations in lung adenocarcinoma. So among the many things these two mutated oncogenes do, one of the shared things they do is signal through the ERK MAP kinase pathway. So this is a very important pathway in normal cells that contributes to survival and proliferation. And this pathway is co-opted by these oncogenes to promote cancer survival and proliferation. So some early analysis by my supervisor, Will, of mutations in lung adenocarcinoma patients found, or Will and others, found that these mutations actually tend to be mutually exclusive. So this is a chart of mutations detected in patients, and we see that there's actually very limited overlap between some of the common oncogenes like KRAS, EGFR, BRAF, and some of these oncogene rearrangements. If we look specifically at mutant KRAS and mutant EGFR in lung adenocarcinoma patients, there's actually almost no overlap. The one case here was actually a non-signaling EGFR mutation. So there's actually significant mutual exclusivity between mutant EGFR and mutant KRAS. So this begs the question, why are mutant KRAS and mutant EGFR mutually exclusive? So my supervisor, Will, went about investigating this using these DOX-inducible promoters, where under the control of DOX, you can express a target gene. And they used these in cultured cells to co-express driver oncogenes together. And what they found was that this led to cell death over time. So if you overexpress KRAS G12V in an EGFR mutant cell line, or if you overexpress mutant EGFR in a KRAS mutant cell line, you got a loss of viability over time. And in mouse, if you co-expressed EGFR and KRAS together, you did get tumors, but you got tumors that only express one of the two oncogenes, never both together. So this begs the question, what drives synthetic lethality between mutant EGFR and mutant KRAS? So some signaling work using microarray and phosphoproteomics revealed that activation or that co-expression of mutant EGFR and mutant KRAS together led to strong activation of the ERK MAP kinase signaling pathway. And so would it be possible that there would be excessive signaling through this pathway, and this could be leading to cell death over time? So the experiment to test this was just a simple rescue experiment treating these cells that are co-expressing EGFR and KRAS together with an inhibitor of MAP kinase pathway, in this case, trametinib, which inhibits MEK. And they found that if they treated with trametinib, they could actually rescue the loss of viability induced by co-expressing EGFR and KRAS together. So this suggests that these cells are sensitive to an excessive amount of signaling through this normally critical pathway. So this is sort of a new vulnerability for lung adenocarcinoma cells that is my main area of study. So we typically think of cells as needing sort of these cells that are either EGFR or KRAS mutant are dependent on very fine-tuned levels of ERK signaling. If they have a single mutation, they're in what we call a sort of Goldilocks zone. If we treat these cells with an inhibitor, such as EGFR tyrosine kinase inhibitor or kinase inhibitor used in the clinic, we can turn the pathway off, and these cells are highly sensitive to this, and this has informed generations of targeted therapies used in the clinic. However, what we have found is that these cells are also sensitive to too much signaling through this pathway, and this has been recapitulated by overexpressing EGFR and KRAS together. So my area of study on ERK hyperactivation has been in the context of drug resistance. So almost all lung adenocarcinoma patients treated with MAP kinase targeted therapies eventually develop resistance to these. On the right here, I'm showing a pie chart of some of the mechanisms of resistance to osimertinib, which is a common EGFR inhibitor given to patients. Many of these mechanisms of resistance, I've highlighted a few here, could potentially reactivate MAP kinase signaling in the presence of drug. So is there potential for ERK hyperactivation in any drug-resistant lung adenocarcinoma cells? And could this be a new way of targeting drug-resistant cells? So we have several models of drug resistance to study this in. One of the models we looked at is a model of trametinib resistance in KRAS mutant lung adenocarcinoma. So trametinib inhibits MEK, and trametinib showed great promise in preclinical models of KRAS mutations, however, failed to translate in the clinic due to the onset of resistance. So we wanted to study resistance to trametinib. So we started with two KRAS mutant lines, H358 and H1792, both lung adenocarcinoma cell lines. And we made them resistant to trametinib by culturing them in increasing concentrations of trametinib over time until they were resistant to 1 micromolar, at which point we considered them resistant. And what we noticed right away is that one of the clones we generated showed this kind of like bell-shaped dose response curve, suggests and seemed to be that some of these cells were more viable in drug than relative to the DMSO treated control. So this is kind of curious, and we decided to investigate this further. And we first validated this with a few growth assays, and we confirmed that these cells actually do grow better when treated with the drug than when treated with the vehicle control. They also show much decreased colony forming potential when we remove the drug. A Western blot analysis revealed that drug removal in this drug resistant cell line actually induced apoptosis, similar to the apoptosis induced when we treat the sensitive parental cell line with trametinib. Yeah, and this is some microscopy images further confirming that these cells really are like they die when we take them off drug, which is really curious. So given trametinib's inhibitory properties on MEK and MEK being right above ERK in the MAP kinase pathway, we wondered, is ERK hyperactivation inducing cell death in this context? So the experiment for this is a pretty simple one. We just did a Western blot where we removed drug and I collected lysates at various time points. And we see that as soon as 30 minutes after I removed drug in this resistant cell line, there's a really, really strong increase in phospho-ERK, suggesting ERK activation that is sustained past 72 hours. And this is correlated with later induction of markers of apoptosis. We also blotted for a few factors downstream of ERK to confirm that ERK was actually signaling and not just, or ERK was actually mediating a signaling effect. And we do see induction of some proteins downstream of ERK, some signaling proteins downstream of ERK. So next, to test whether or not ERK is killing
Looks like we might have lost Dylan.
Yeah, that's possible. The Wi-Fi at BC Cancer is not ideal. Let's just give him a minute.
Yeah.
And hopefully he can pick up where he left off.
Yeah, I don't see him in the participants list anymore. I think let's move on with the next talk. And then if Dylan comes back, he can pick up where he left off. Oh, Kathy, you're still muted.
Let's give him a minute.
Okay. Oh, there he is.
There we go.
Sorry about that. I'm on edgy roam and it's been very shoddy lately.
No worries. We feel the pain.
Yeah. Okay.
If you go one more slide back.
Sure, yeah. Okay, where did I cut off?
You cut off, you had just introduced the first confluency graph.
Okay. Yeah, so like I was saying, basically to see if ERK hyperactivation is killing these cells, we just tried to rescue them by treating with an ERK inhibitor. And in this assay on the left here, we see a complete rescue of cell growth when we treat with this ERK inhibitor. And in the Western blot on the right, we see that we can rescue both the induction of phospho-ERK as well as the induction of these markers of apoptosis by treating with an ERK inhibitor, suggesting that ERK is responsible for mediating cell death. Next, we wanted to investigate whether specifically ERK1 or ERK2, because ERK exists as a pair of isoforms, was mediating this phenotype. And very interestingly, it was ERK2 specifically that was killing cells. We typically think of ERK1 and ERK2 as having pretty overlapping functions, but in this specific case of mediating cell death, there's a really clear distinction between ERK1 and ERK2, which is kind of curious and something we haven't fully figured out yet. It was very interesting. But yeah, based on this, we figured out that, ERK hyperactivation is killing these cells, but what is actually inducing ERK hyperactivation? What is upstream of ERK? What is mediating this excessive amount of signaling after we move trametinib? So to figure that out, we submitted our cells for next generation sequencing at this MSK impact. We submitted our cells to this MSK impact panel for sequencing. And among the many alterations that we detected amplifications in these 4 genes that are known to activate MAP kinase signaling. So could one of these 4 genes actually be responsible for drug addiction? We confirmed the amplification at the protein level. And if we look at our cells overall now, we have amplification of proteins at 3 different nodes of the MAP kinase signaling pathway. So any one of these could, in theory, could be driving drug addiction. We wondered if KRAS G12C amp was driving ERK hyperactivation following drug removal. So to test this, the experiment, again, is pretty simple. We just treated our cells after drug removal with a newly developed inhibitor for KRAS G12C. And we see that it actually rescues the cells pretty well. Again, by Western blot, we see that treatment with AMG 510, the KRAS inhibitor, rescues the induction of phospho-ERK, as well as partially rescuing the induction of these markers of apoptosis. We were wondering if it was KRAS G12C specific, like the mutant KRAS amp specifically, or total KRAS. We tried to rescue these cells by knocking down KRAS with siRNA, and we saw that this actually did not rescue our cells and actually further sensitized them to trametinib. So this suggests that KRAS amp might be responsible for driving resistance to trametinib, but total knockdown of KRAS did not rescue. Since total knockdown of KRAS did not rescue drug addiction, it was not mediating drug addiction. Next, we wanted to validate or to see if any of the other genes that were amplified were driving drug addiction. So we just did a bunch more rescue experiments and confirmed that none of these other proteins that were found to be amplified were driving drug addiction. So it's really dependent on KRAS G12C amp and ERK. So to recapitulate this phenotype, we overexpress KRAS G12C in KRAS mutant lung adenocarcinoma cell lines. And we see that after 24 hours, we can reproduce the induction of phospho-ERK. And after 9 days, we see a loss of viability. And we confirm this in H358, which have a single mutation KRAS G12C, as well as H23 and H1792, 2 more KRAS mutant lung adenocarcinoma cell lines. And we also confirm that we can rescue the loss of viability induced as well as the induction of phospho-ERK by treating with either AMG 510, the KRAS G12C inhibitor, or trametinib, our MEK inhibitor. So in summary, what I've shown you is a cell line that is initially highly sensitive to trametinib. We see that when we treat with trametinib, we get induction of apoptosis. After a long-term culture in trametinib, these cells become resistant through amplification of KRAS G12C. And this mechanism resistance is only tolerated in the presence of trametinib. As soon as we remove trametinib, we get this strong induction of signaling through the MAP kinase pathway, which results in hyperactivation of ERK, which also leads to cell death. So some of the future directions for this work, we're also looking to investigate, see if there's any evidence of drug addiction or ERK hyperactivation in the context of osimertinib and sotorasib, which are osimertinib is an EGFR tyrosine kinase inhibitor that is currently given to patients. And sotorasib is a KRAS G12C inhibitor, which is also being given to non-small cell lung cancer patients. So as with many targeted therapies, resistance to these 2 therapies are frequent occurrences. And we want to investigate if any of these mechanisms of resistance might reactivate MAP kinase pathway and lead to ERK hyperactivation. So with that, here are my acknowledgements. And yeah, thank you for your time.
All right. Thank you so much for that great presentation, Dylan. It's obvious to see why we call you the Western Blot Wizard. So I will open up the floor now for any questions. Feel free to raise your hand or drop them in the chat and I will unmute you. All right. All right, why don't I start us off here? So Dylan, is there any chance that this exclusivity in mutations is not just towards lung adenocarcinoma? Are there any other cancer subtypes that you might want to explore as having this associated mechanism in the future?
Yeah, that's a good question. So some of this work similar work has actually been done in melanoma.
Okay.
And the common, like melanoma often sees, like one of the driver mutations in melanoma is BRAF V600E.
BRAF, yeah.
They get treated with BRAF inhibitors, and one of the mechanism of resistance is amplification of BRAF V600E, and they actually observe similar effects in melanoma, where as soon as you remove the BRAF inhibitors, you get like hyperactivation of ERK and cell death. So it seems to be, there's some work that has characterized this in melanoma. I don't know about any other cancer types.
That's very interesting. All right. Any other questions? All right, I guess that we're going to move on to our next speaker. So Nishadh, if you don't mind sharing your screen. All right, thank you so much. So our next speaker is Nishadh Rathod, and he is a PhD candidate at the University of Alberta in Dr. Howard Young's lab. And today he will be presenting his project entitled Regulation of the Sarco-Endoplasmic Reticulum Calcium Pump by Phospholamban, studied using in vitro functional studies and in silico alchemic free energy simulations. And with that, take it away.
Oh, you're still muted, Nishadh.
Yep. Oh. You're still muted, Nishadh.
Oh, I see. Okay.
There we go.
Great.
We can hear you now.
Awesome. So thank you for the introduction. So today I'll be talking about regulation of calcium pump, SERCA, by its inhibitor phospholamban. Okay, so SERCA belongs to this P-type ATPase family. So one of the popular P-type ATPase pump you probably almost have heard is the sodium potassium pump. But there are also other kinds, such as the SERCA, which is the calcium pump, as well as the proton pump and zinc copper ATPases, and more famously phospholipids, lipases, and phosphases. And there are also this new family of P-type pumps, that those are the polyamine pumps. But today I'll be mostly talking about this calcium pump, P2B. So there are 3 genes that code for SERCA, SERCA1, 2, and 3. SERCA3 is mostly expressed in non-muscle tissues. SERCA2 is ubiquitous and also to some degree in cardiac and skeletal muscles. In particular, SERCA1A that has this 1A isoform expressed in the skeletal muscle, which is functionally equivalent to the SERCA2A, which is expressed primarily in the cardiac muscle. So if you look at the heart muscles of the heart, there are these myofibrils that contain the web of the Sarco-Endoplasmic Reticulum. And if you look at the interface of the SR and the cytoplasm, we see 3 important proteins. So these include SERCA, obviously, randin receptors, and the L-type calcium release channel. So whenever there is an electrical signal from the SC or AV node, the electrical current leads to the release of this calcium from the SR into the cytoplasm using this via this randin receptor, which results in the contraction of the muscle. The opposite happens during the contract relaxation, where the SERCA is actively pumping calcium from the cytoplasm and into the SR-ER. So as I mentioned, SERCA is regulated by this inhibitory peptide called phospholamban. So if you look at some of the structural details, the cytoplasmic domains of SERCA include this nucleotide binding domain where the ATP molecule binds. And there is this phosphorylation domain where it phosphorylates a aspartic acid. And as a consequence of that, it results in this movement of this actuator domain that functions to open this channel for the transmembrane that allows movement of the calcium from the cytoplasm into the lumen. So the inhibitor phospholamban, it could exist as a monomer or a pentamer. So it is in this dynamic equilibrium between these monomers. So again, phospholamban is also expressed in the cardiac muscles, and some of the mutations in phospholamban have been implicated in the various different heart diseases. So these include hypertrophic, restrictive, dilated, and even ARVC cardiomyopathies. So previously, there has been a structure for this complex involving SERCA-bound to phospholamban have been identified, but only the transmembrane region of it has been resolved using X-ray crystallography. So in the first part of this project, we wanted to look at how each of the alanine mutations in phospholamban result in the inhibitory activities of regulating the inhibitory activities of. So obviously we started off with PCR, then expressing these peptides in E. coli, and then we carry out this affinity purification and subsequent HPLC purification. And once we have this purified peptide, then we reconstitute it into the vesicle containing SERCA as well as the peptide. So if you look at the microscope image, the vesicles look something like this that contain SERCA and phospholamban with that. And then we go on to collecting some of the functional activity data that gives us indication of how these alanine mutations result in the inhibition of the pump. So here is a typical functional data. So if you look at the negative control that contains only SERCA in the vesicle, which has a kCa of around 0.46 micromolar, versus the positive control that has the wild type phospholamban plus the SERCA. So this has a rightward shift indicating a very strong inhibition, whereas as an example, another mutation such as the R25 mutated to alanine has a relatively lower rightward shift, so it has a Kc around 0.56 micromolar. So it could, from this, it could be stated that it is a partial loss of function mutation in phospholamban. So we carried out alanine mutation all the way from microleucine 18 to LL52 to alanine in phospholamban, and we then characterize each of these mutations as being superinhibitory or loss of function. So a lot of these mutations are in the cytoplasmic or the linker region of the phospholamban are partial loss of function, whereas the transmembrane region has a predominant superinhibitory effect. So the ones highlighted in green are the superinhibition or gain of function mutations, versus the ones in orange are the loss of function. So hold on to this part of the experiment. I'll come back to this. So in the part 2 of the project, then we looked at how the affinities between phospholamban and SERCA are being affected upon mutating to alanine. So for this, we used the in silico method of the alchemical simulation. So I won't go into much detail just for the interest of the time. But the general concept here is that we carry out 2 sets of different simulations, one in the unbound state and state. So the top row corresponds to the wild type finding or the delta G of the wild type finding, versus the bottom row corresponds to the delta G of mutant finding. So in this case, it would be each of these alanines, each of these residues mutated to alanine. So in the unbound state, when we determine the difference in the 3 energies for mutations, and then we compare that in the bound state, it gives us the difference between the how it compares the delta G of formation between mutant versus mutant. So from this, we can characterize how each of the alanine mutations the calculations including the. So from this, the whole objective is to determine the probability distribution for each of the mutations that we did, and when we get this flat line in each of these probability distributions, it indicates that our values have been converged, and from that point on, we can just move on to determining the delta G. So in this example, glutamate 19 mutated to alanine in phospholamban equates to about -4.89 plus minus 0.28 kcal per mole. So similarly, we determine the delta G for all the mutations. So some of them increase formation corresponding by the negative delta G, versus some of them decrease the formation of the SERCA-bound complex corresponding by the positive values. So here we highlighted the increased binding affinity with the green and the decrease with the orange. So how does this delta G that we determined from the in silico compare with the delta KCA that we determined from the functional? So on the x-axis, we plotted the delta KCA, and on the y-axis, delta G. So on the mutations on the top left quadrant correspond to the less formation of the bound complex, which results in the loss of function, and the bottom left quadrant corresponds to the mutations that increase the formation of the complex, but yet are loss of function. So this could be explained by the mechanism that upon binding phospholamban, when it binds to SERCA, it fails to affect the 2 calcium binding sites, which are in direct opposite to this M2 and M6 helices. However, it fails to explain all these superinhibitory mutations, especially the ones that are significantly superinhibitory, seems to have a very less correlation with the delta G. So then we went on to repeating the same delta G, but in the pentameric form instead of the monomer. So the difference here is that the monomer was in completely membrane environment, whereas the pentamer is obviously each of the protomer is in the environment of the other 2 that are in the adjacent in the pentameric state. So here we can see that all of these superinhibitory mutations have a really strong correlation with the delta G and the delta KCA. So what this probably indicates is that the bound complex with SERCA and phospholamban probably forms from both monomer and the pentamer. So, and just to give you a visual summary of this, so obviously phospholamban is present in the dynamic equilibrium between pentamer and monomer, but it can bind to SERCA as a monomer as well as it bind directly from a pentamer. So when it binds directly from a pentamer, it results in a superinhibitory effect. This is usually the case when it is mutated dwelling, which increases this pentameric to the Sarco-Endoplasmic complex formation path. So if you look at some of these structural details, the ones that are basically loss of function are pointing, these residues point directly towards Sarco versus the ones that are gain of function or the superinhibitory are directly away, pointing away, and these probably participate in the interaction with the pentamer. So these are the interactions that are weakened upon mutating to the element. So with that, I would like to acknowledge everyone in my lab as well as my supervisor, and especially this CSMB student committee for organizing this wonderful seminar series. Happy to answer any questions. Thanks.
Thank you so much, Nishadh, for that wonderful talk. And again, if there are any questions, feel free to write them up in the chat or raise your hand, and we'd be happy to unmute you. All right. We have our first question, I believe. Feel free.
Yeah. So my question is in the last slide. If you can go back to the last slide. Yeah, here. Yeah. So I see that all the gain of function are also like hydrophobic, and the others are also like both sides it's hydrophobic. So I'm not able to understand the explanation here. Like, why is like one giving you gain of function and others loss of function?
Okay, so it's primarily the pentamer, right? So the ones that are in red, these form the leucine zipper that is involved in the pentameric, stabilizing these pentameric interactions. So when you mutate them to alanine, this pentameric interaction is weakened so that it can directly form a complex with the Sarco. So when you weaken this pentameric state, it can form more of this complex. So if you look at it in terms of just the energetics, this pentamer would be like the most highly stable pentameric species in the membrane compared to the Sarco-Endoplasmic complex. So when this highly stable species is weakened, it frees up more of this available phospholamban for it to go and bind to PLN. So that's why we see this differentiatory effect.
Yeah, so this is from the experiment and not from the MD simulation, right? Because in the MD simulation, when you do it as monomers, you would not see any change then.
We actually correlated both MD simulation and the functional studies. So we correlated both with the in the monomeric as well as the pentameric. So if you look at this is strictly from the pentameric versus this one, which is from the monomeric. So on the x-axis is the functional data versus the y-axis, which is the in silico strain energy calculation.
Yeah, so in this case, all the residues that are facing on the outside in the monomeric form, mutating them should not have changed your affinity at all, right? Because it's in the monomeric form and it's not interacting, so the affinity should not be changing.
So for these ones, right, the ones that are on the
facing away from the interface. Yeah, those that are facing away from the interface in the monomeric form should not have any effect on the affinity values, correct? As per your point that you make, that because those are in, but I do see it different, right?
Sorry, I don't understand your question properly. So what residues are you trying to say?
So the residues that are facing away in the monomeric form, if you mutate them in the monomeric state in your MD simulation, do they affect your affinity values or not? That's my question.
No, not to a greater degree. So this is directly indicated here. So the ones that are on the x-axis, exactly on the x-axis, especially these two residues, they don't affect the affinity at all, like to a very minimal degree, versus these ones, which affect the affinity to a much greater degree. So these are the same ones, same residues, but in the pentameric state, you see a much greater or negative delta G.
Okay.
So the
Thank you.
KCA values are exactly the same in both graphs.
Okay. Thank you.
All right. Go ahead, Ma.
Really nice talk. That was really cool work. Thank you. I just had more of a clarification question. I was curious, is it known that the phospholamban forms a pentamer, or is that just inferred from the simulation data?
No, it does form a pentamer.
Okay.
So there are a few structures up there that also show there is a pentameric association of phospholamban, and this has been well known that it forms its pentamer due to the leucine zipper.
Okay. Thank you for the clarification.
Yeah.
Right. I don't think there are any more questions. All right. So with that, thank you so much, Nishadh and Dylan, for some really great talks today. And stay tuned for emails and some Twitter advertisements for our next CSMB seminar, which will be taking place in July. With that, I hope everyone has a great day and see you later.